Hello and welcome to Nikolai's genetics lessons, and today's first question is: molecular sequence data has been shown to be more reliable than morphological data for estimating phylogenies. And what you can see on this picture, of course, you can say that just by comparing appearance of the humans and chimps, for example, we can tell that they are more closely related than, for example, humans and mouse, because just by their morphological features. But sometimes it is hard to tell when we would compare some organisms that are closely related, for example, other great apes in relation to humans and themselves. In this case, molecular sequence data would be the most reliable source of information for us to build phylogenetic trees like this. So this is true statement. And next question: what is the genetic distance between human and chimp as shown in the tree below? And here's a reference fragment which represent number of substitutions per site per certain amount of time. Some of you may think that our logic have to be as follows. So here's a point where these two species separated, but here at this particular point in time they were one species. So we just have to count how many such fragments we have on each branch. For example, 1, 2, 3 here, 1, 2, and 3 here. So our answer have to be 0.06 substitutions per site. Let me add to this picture a timeline. So here is going to be our timeline, and now if we project these horizontal lines here, we would see that they would take certain fragments on this timeline. And how much time would take this vertical line? It actually is not going to take any time at all. Only horizontal lines would represent certain amount of time here and not vertical ones. So when we do our calculations, we ignore these vertical fragments. So now you also may face a choice of two variants. For example, so this is going to be how we project a time here, and this is going to be two fragments here like this. Should we choose then, in this case, this variant D as an answer? If, for example, chimps would die at this point as species, then humans would evolve and would be separated from this point in time by these two fragments and would accumulate certain number of mutations if we compare, for example, DNA sequence or protein sequence. But because these two species evolve independently, both of them, both these lines, would accumulate over the period of time certain mutations per site. It can be mutations on the DNA level, or we also can compare, for example, protein sequence. So in this case, we have to add this time and this time, and we would say that these two species would accumulate 0.01 mutation per site here for this period of time, 0.01 mutation here, and 0.01 mutation here, and 0.01 mutation here. So each species here would accumulate 0.02 mutations per one site for this period of time. But these two species, when we compare, we have to add these numbers. So that means that they would be separated, these two species, by 0.04 substitutions for this period of time. This is answer B. But what if we want to compare, for example, how humans and rats related? Then in this case we have to take into account this distance here, here, and here. And this is all for today. Subscribe and see you in the next video. Goodbye.